Doctors and professors, thank you very much for attending the APASL 2024, the Center of Hepatology. Now, we'd like to start our conference program. First session, HBV, one challenges for elimination of CCC HBV DNA, chaired by Dr. Yoshiyuki Ueno and Dr. Shuhei Hige. Now, Doctor, please.
Okay, members and guests, this session is very hot topics, and unfortunately, my co-chair, Professor Jin Jia, couldn't make this session today, and instead of him, Dr. Shuhei Hige, who was supposed to be a discussant, will serve as a co-chair with me, and we move to the first speaker. Clinical efficacy of novel biomarker for hepatitis B infection from MF Yuan. Everybody knows Professor MF Yuan, and I'd like to invite him as podium. Thank you, MF.
Thank you very much, Chairman, and thank you, the organizer, for inviting me to talk on this specific topic. I understand that the topic is the challenges for CCC DNA elimination, but instead I want to give a talk on the novel biomarkers of hepatitis B, because we know that measuring CCC DNA requires liver biopsy. So it will be really difficult in clinical practice where we can have an idea of the CCC DNA content in the liver. So instead, I try to talk something that may reflect the CCC DNA content in the liver. So I chose to talk two specific novel biomarkers for hepatitis B infection. So these are my disclosures. So when we talk about viral markers in the blood, we have the conventional ones, including surface antigen, E antigen, and HBV DNA. When we are talking about novel biomarkers, we have what we call p-genomic RNA and also correlated antigen. And this diagram shows you in the blood that we can measure the DNA, surface antigen, the RNA, the E antigen, and the collectively correlated antigen composed of the core antigen, E antigen, and other subunit of P22CR. So when you look at the hepatitis B genome, because of the overlapping four with overlapping genes, and that you can get what we call different RNA transcripts. We have what we call p-genomic RNA, we have p-core mRNA, we have surface mRNA, and also xmRNA. So accordingly, so if you look at the, you transcribe or translate the p-core RNA, then you will get the E antigen. Since E antigen is a component of the core-related antigen, so it will be reflected as a core-related antigens. If you have the transcription of surface antigen, then obviously you will measure the surface antigen. This xRNA will be, protein will be in the liver, and at the same time, if you have a p-genomic RNA replications, reverse transcription, then you will have p-genomic RNA, the HBV DNA, and also the core antigen. And as I said, the core antigen is one of the components of the core-related antigen, so it reflects as a core-related antigen in the blood. In fact, in patients with hepatitis B, when you measure the blood for different components of the virus, there are 5 already. If you measure if you take a look on the complete variants in the blood, you can not measure the RNA because all will be the DNA, capsid, core protein, and the surface antigen. We know that we have RNA variants in the blood where you do not have the HBV DNA. We also have something called empty variants where you don't have any nucleic acids. So in that sense, you will measure the surface antigen and core-related antigen. We also have what we call linked capsids, where you will not be able to measure the surface antigen, but you can measure the nucleic acids and also the core protein. So viral particles is very famous, all right? Nothing but only the viral proteins. Majority will be the surface antigen. So what is core-related antigen? It's actually a collective measurement of 3 different units or different biomarkers in the blood. So if you have the genomic RNA, then you will have complete virions and empty virions, and that will constitute the core antigen. When you have the translation of a P-core mRNA, you will have E antigen as well as a P22CR. So these 2 together with the complete virions where you can detect the core antigen that will collectively being measured by what we call core-related antigen. And in fact, when we look at all the biomarkers in the blood, including the surface antigen, E antigen, core-related antigen, HBV RNA, they all follow the same pattern in the different phases of chronic hepatitis B infection, meaning that we got all the biomarkers at the highest level in the E positive phase, particularly higher in the E positive infection. And then seems to be lower in the E negative infection phase. But then if you have E negative hepatitis, all the markers actually gone up. And then if you're lucky enough, you have surface antigen zero clearance, then all the markers will be decreased. And in fact, when we look at this particular group of patients, we can still measure some, positivity by measuring the core-related antigen and HBV RNA. According to a study, at least 16% of those who lost a surface antigen, you still can have the detection of either one, core-related antigen or HBV RNA. And in fact, when we look at the core-related antigen, after patients achieve functional cure, you can see some of them, they still have a measurable core-related antigen even after 8 years of losing surface antigen. How about the detectability of these 2 markers in different groups of patients? When we look at treatment-naive patients, E positive, both markers will be positive, 100%. And then when you look at E negative patients, treatment naive, you can see if you have chronic infection and chronic hepatitis, then the RNA will be around 80% to 97%. A little bit lower for core-related antigen, 70% to 88%. However, when we measure these 2 markers in NUC-treated patients with undetectable DNA, then the RNA still have a reasonable detection rate, but then we get a significantly lower core-related antigen in this particular patient. So the application of core-related antigen in this particular group of patients who are on nucleoside analogs with undetectable DNA, the detection rate is not optimal. Because of this, the company actually developed a more sensitive assay. What I showed you in the slide previously, we use what we call conventional, or the first version of a core-related antigen with the sensitivity of around 3 log unit per mil. With the more sensitive assay, they call it ITAC, with some pretreatment on the sample, disrupting the disulfide bonds, the sensitivity can be as low as 2.1 log. And this really make difference. If we use the convention of the first generation core-related antigen to measure in patients who had surface antigen zero clearance, we can measure around 21%. So 21% after 1 year of surface antigen loss, we can have the measurable core-related antigen. But then, if we use a more sensitive assay, after 1 year of a loss of surface antigen, 2/3 of the patients, you can still detect this core-related antigen. And this slide shows you the profile of all these patients. In fact, when we look at the ITAC, the more sensitive assay for core-related antigen is actually around 65%, even after 10 years of losing surface antigen. Whereas if you look at the even using a highly sensitive surface antigen level test, it's only 24%. So you can see over the years, from the time of surface antigen loss till 10 years, they actually have a very consistent detectability rate in these patients, unlike using the ITAC version for surface antigen. It drops in terms of the detectability rate, while the antibody receiver conversion increases. How about the treatment effects of these two markers on this two novel markers? This is a very simple diagram showing you that entecavir, tenofovir, or TDF or TAF, after being treated for 48 weeks, most of patients, they actually have a very profound DNA suppression. How about COVID antigen? How about HBV DNA? So for COVID antigen, there was also continuation of the reduction upon 48 weeks and 96 weeks of therapy. This is the RNA. So very similar, all right? Baseline, 1 year, and then 2 years. So if you treat the patient with entecavir or tenofovir, then you will expect drops or reduction of a COVID antigen as well as the RNAs. Even when we separate the patients into E positive and E negative, you can see there was no significant difference in terms of the reduction of the COVID antigen in patients receiving tenofovir-based or entecavir-based therapy. Same for the E negative patients. If you prolong the treatment for 7 years, you can still see a continuation of decrease or reduction of COVID antigen with the median reduction over 7 years of 2.2 Ku per ml. How about HBV RNA? So this is an early study showing that after 1 year of treatment, you see, as expected, very profound DNA suppression, minimal effect on surface antigen, but you see I would say modest effect on the RNA after 1 year of treatment. We also try to look at the effect of 2 years treatment, the median drop or the mean drop of the HBV RNA after 1 year was around 1.5 log, and then continued to drop to 1.8 logs after entecavir treatment for 2 years. But mind you, in fact, at least 78% of these patients even have a drop of surface antigen, RNA. They still have the measurable, quantifiable RNA in the blood. This slide just, repeat the one that I have shown you. I just want to illustrate that the magnitude of reduction of these two markers are very similar. So you will expect the same log reduction when you treat the patients with nucleoside analogs in terms of the RNA and as well as the COVID antigen. How about the effect of novel therapy on these two markers? And this is a study using the ASO, bepirovirsen, to look at the effect on the surface antigen, HBV RNA, and COVID antigen. So for E negative patients, they actually show a very good reduction of the RNA as well as the COVID antigen in E negative nucleoside or treatment naive populations. And the RNA and COVID antigen reduction maintained even after the treatment. How about siRNA? So this one is one of the siRNA being studied, and you can see this is a very, demonstrable reduction of the COVID antigen after 4 doses of this siRNA, and after stopping, the level actually plateau or sustained for at least 1 year after post-treatment. Same for the RNA. You can see a very nice drop of RNA upon the treatment with siRNA, and then maintain at this particular level even after stopping of therapy. Another agent, siRNA, showing the same thing. E antigen reduction, plateauing effect or sustained effect, COVID antigen reduction, sustained effect, as well as the surface antigen. Mind you that this study only gave 3 doses of this J&J 3989. V2218, you can see this is for 50 milligram, this is 200 milligram. You can see those proportional response in terms of the COVID antigen reduction, as well as E antigen reduction and RNA reduction, and maintain after off therapy. Imdusiran, another sRNA, showing you the same thing, even at a single dose or multiple dose, then you can see some patients with very nice drop of the RNA level as well as the COVID antigen. And finally, I just want to bring your attention to this particular study, looking at examining the effect of the CAMs on the effect on the DNA, RNA, and surface antigen. And this is a 15E positive patients, treatment naive, part 4 cohort 2 patients. When you have the patients just receive the entecavir, then you expect a drop of DNA. But when you look at the patients who receive CAMs, actually, together with entecavir, you have a bigger drop of the HBV DNA. When you look at the RNA, very low, modest or mild drop of the RNA. But then if you have the patients with 2 therapy together, then you have a very nice drop of RNA. One thing is, for those who are on entecavir after 12 weeks, with the addition of the CAMs, you can see there will be a nice drop of the RNA too. When we look at the surface antigen, this is the first CAM ever studied till now, have a very nice surface antigen reduction. And this is another cohort using 300 milligram, and you can see a very nice reduction of DNA maintained throughout the treatment. And more importantly, you can see this reduction, individual plot of the DNA reduction. The most important effect of this particular effect, spectacular effect of this antigen, this compound, is the ability to lower down the surface antigen, E antigen, and COVID antigen. In contrast to nothing happened in patients who are on entecavir monotherapy. So my conclusion is COVID antigen and RNA are easily measurable in the blood. CCC DNA transcriptional activities may be represented serologically by the core-related antigen and RNA, whereas the surface antigen generated from the CCC DNA and integrated DNA. ITAC core-related antigen has a better detection rate in patients with surface antigen seroclearance. HBV RNA has a better detection rate in NUC-suppressed patients. Both serve as biomarkers for target engagement or proof of concept of the different modes of action of novel antiviral agents. And I think combining these two assays to improve disease progression, prediction, prognostication, treatment response in hepatitis B cure program will be highly valuable. With that, I will thank you for your attention.
Thank you, Professor Yuan. And this talk is open for discussion. Any questions, suggestions from the audience? Okay, I'm just asking about the technical issues. The measurement of HBV RNA, is it equally difficult to measure H-core-related antigen or something?
I'm sorry, can you say?
Technically, it is same
For
same difficulties. Technically, it's only it's like PCR, quantitative PCR. It is very...
the RNA.
Yeah.
In fact, it's actually it's a PCR assay. It's actually a reverse transcription assay. So it's actually technically not too difficult. But then people are now, fine-tuning the sensitivity level for the RNA.
So it can use in a very practical setting, in daily clinical settings.
Yes.
Okay.
Yes.
Any questions? Yes, please.
The effect of interferon on these two markers? No. I think there may be 1 or 2 studies, but not too well studied. Too many studies on using interferon on these 2 markers. But I would suppose to see a reduction too in terms of the RNA as well as the COVID antigen. But the magnitude of reduction, we don't know.
Yeah. Okay.
At first, I'd like to ask you about HB core related antigen positivity with other HBV marker negative S antigen and so on. And
in many studies, previous study, we have shown that there was a very good correlation between the COVID antigen, serum HB DNA, intrahepatic DNA, CCC DNA. So COVID antigen actually has a very good correlation with different markers, either in the serum or even in the liver.
And it's just owing due to sensitivity.
The sensitivity improved, as I said, okay, from 3 logs to 2.1 logs. So that will improve the detection rate in different situations.
Okay, and then next, I want to know, could you give me your thought about a comprehension of evaluation of HBs antigen in clinical settings? I think HBs antigen amount accurately correlate, reflect CCC DNA when HBs antigen was tested in a laboratory laboratory setting and using, for example, hepatocytes or mice. But at that time, integrated DNA not included in that situation. But in clinical setting, those the correlation between CCC DNA and S antigen decreased when the status of HBe antigen changed from positive to negative. At that time, the origin of HBs antigen also changed from CCC DNA to integrated DNA. So how should we comprehend the S antigen level, especially in case of HBe antigen negative?
We understand all this because, as I mentioned in the conclusion, the surface antigen actually can come from the CCC DNA as well as the integrated DNA. This is a problem of how to interpret the surface antigen in the blood. All right. So if you measure the surface antigen in the E positive patients, you don't know how exactly the surface antigen reflect the CCC DNA. But then if you measure the surface antigen in E negative patients, you will presume that this surface antigen is likely to be coming from the integrated DNA, although it's not absolute. Yeah. So in general, if you really want to correlate the CCC DNA contents, it has also been shown in our study, a surface antigen correlation is not better. In fact, it's less effective when you compare the correlation with COVID antigen and RNA. Yeah. So these are the more, sensitive markers to reflect CCC DNA because surface antigen have another source of, from the integrated ones.
Thank you.
Oh, yes.
Yes.
Hi, MF.
Hi.
Great talk. Could I ask about CAMs? Now, these CAMs will alter the structure of capsids, right? Have you done the analysis of whether these altered forms are still detected by the by your correlated antigen?
This is an excellent question. If you use CAM, okay, as I said, it affects the assembly of the capsid. The capsid. I don't know whether we can still measure this altered capsid in the circulation. And just like the one the background slide I showed some capsid capsid, empty capsid in the blood. Theoretically, yes, but no one actually did the measurement.
Yeah. So I'd imagine with your new method where you're denaturing everything with urea and acid, that might help. But
sure,
yeah,
definitely.
Okay. Thank you very much.
Thank you.
We'll move to the second talk entitled as Identification of Host Factors that were essentially involved in hepatitis B virus persistence by Professor Masao Honda. He's give a talk through the web. Please go ahead.
Yeah, okay, thank you. All right, thank you, Chairman. Today, I would like to talk about identification of host factors that are essentially involved in hepatitis B virus persistence. So hepatitis B virus HBV infection is difficult to cure owing to the persistence of covalently closed circular viral DNA, CCC DNA. So it is generally understood that HBV enters the cell through HBV receptor NTCP, and then the virus receptor complex is encapsulated in early endosome and passed to the late endosome, where the virus particles are exposed to the lysosome enzyme and capsid protein, including a viral genome, escaped from the late endosome and go into the nucleus. So today, I'm going to talk about the interesting findings that we have. Firstly, there is another retrograde trafficking route from early endosome to the Golgi and endoplasmic reticulum that facilitates HBV entry into the nucleus. And secondly, DOC11, a dedicator of cytokinases 11, is involved in this retrograde trafficking of HBV. So in the beginning, we newly established HC1 cell that is isolated from resected HCC tissues of chronic hepatitis B patient, where the HBV replication is maintained only a limited number of small cells. So we performed single-cell RNA-seq analysis of HC1 cells and found DOC11, a dedicator of cytokinases 11, was overexpressed in HBV positive cell. So we confirmed that overexpression of DOC11 in HepG2 NTCP4 cells increased CCC DNA, whereas in PXB cell, ablation of DOC11 significantly reduced the CCC DNA. So interestingly, in the liver tissue of chronic hepatitis B, DOC11 expression is significantly correlated with the gene of the actin and microtubules and Golgi apparatus. So therefore, we hypothesize that DOC11 could be involved in the HBV trafficking in hepatocyte. So to clarify the cellular localization of DOC11, we established DOC11 inducible cells based on a HepG2 NTCP4 cell using a tetron system. DOX-induced cells showed that DOC11 was overexpressed mainly in the cytoplasm and partly in the nucleus. So immunofluorescence analysis showed that DOC11 localized in the LAB5 positive early endosome and TGN46 positive trans-Golgi network and PDI positive endoplasmic reticulum, as indicated by the yellow color. So it has been reported that retrograde transport from endosome to trans-Golgi network so divert proteins and lipid away from the lysosome degradation. So it is reported also that Shiga toxin, cholera toxin, and human papillomavirus 16 utilize the retrograde trafficking route from early endosome to the ER or nucleus. So these observations prompt us to investigate whether dog11 maintain CCC DNA through retrograde trafficking route to avoid HBV degradation. To visualize the trafficking route of HBV in living cell, so we utilized labeling system of HBV core protein by using a tetracysteine tag sequencing. So tetracysteine tag sequence was introduced into the 155 amino acid sequence of HBV core protein. And then fluorescent Li-Ash dye attached to the tetracysteine tag. So the HBV core protein could be labeled with a fluorescence signal as shown in the slide. So we analyze the time course of the HBV infection by super resolution fluorescence microscopy. So red color indicate the Li-Ash labeled HB core antigen, and green indicate that the early endosome marker Lab5 and the late endosome marker Lab7 and Golgi apparatus, respectively. So white color indicates that co-localization of HB core antigen and these endosome or Golgi markers. So after infection, unexpectedly, we could not detect core protein in a Lab5 positive early endosome, whereas it accumulated in Lab7 positive late endosome. Interestingly, core protein also accumulated in Golgi apparatus. So we next labeled HB surface antigen using a tetracysteine tag labeling system. Tetracysteine tag was inserted into the PS1 region, as we reported in NanoLook reporter HBV particle in hepatology communication previously. So Li-Ash dye attached tetracysteine tag and HB surface antigen are labeled, could be labeled as shown in the slide. So in the time course analysis of HBV infection, HBs antigen in fact co-localized with Lab5 positive early endosome. And then it also co-localized to Lab7 positive late endosome. However, HB surface antigen did not co-localize with Golgi apparatus. So the results summarized in this slide. So HB core antigen could not be detected in early endosome, probably because HB core antigen is encapsulated by the HB surface antigen in early endosome. So for supporting this finding, HBs antigen was co-localized to be detected in the early endosome in HBs surface antigen labeled HBV infection experiment. Time-lapse experiment of HBV infection clearly showed that in addition to the Lab7 positive late endosome, HBV could be transported to the Golgi apparatus. Interestingly, HBs antigen may not be transported to the Golgi. Maybe only HB core antigen could be released from the early endosome to the Golgi apparatus. So in previous study, it has been reported that HBV particle enter the early endosome and processed to the later endosome by Lab7. No report has been found that HBV utilized retrograde trafficking from early endosome to the Golgi. So to examine the importance of Lab7, we established a Lab7 knockout cell. So interestingly, HBV infection was more enhanced in LAB7 knockout cell. So we performed time-lapse analysis with core protein labeled HBV infection. Red color indicated HB core antigen and green color indicate Golgi and ER. So it was found that HBV core protein was transported to the Golgi or ER, and interestingly, accumulation was more enhanced in LAB7 knockout cell compared to the control cell. So these results so far show that in addition to the canonical route, there is the non-canonical retrograde trafficking route from early endosome to Golgi network, an endoplasmic reticulum that facilitates HBV entry into the nucleus. So the key question was how DOC11 is involved in retrograde trafficking through Golgi apparatus. So we examined the effect of DOC11 on HBV infection in LAB7 knockout cell. In LAB7 knockout cell, the expression of DOC11 was modestly increased, and the level of the HBV DNA and CCC DNA were increased in LAB7 knockout cell. And interestingly, knocking down of the DOC11 strongly reduced the level of the HBV DNA and the CCC DNA. So therefore, DOC11 could be involved in the retrograde trafficking of HBV through Golgi apparatus. So DOC11 is a guanine nucleotide exchange factor, so-called GEF. But the functional role of DOC11 for trafficking had not been reported. So we therefore try to identify the responsible molecule that interact with DOC11 for HBV retrograde trafficking. So we examined Golgi fraction from DOC11 stable knockdown cell and performed LC mass analysis. So we identified trafficking associated protein AGAP2 interact with DOC11. It is reported that AGAP2 could regulate retrograde transport from early endosome to trans-Golgi network. So we find that overexpression of DOC11 increase the expression of AGAP2 in a dose-dependent manner. Moreover, suppression of AGAP2 by siRNA significantly reduced CCC DNA in LAB7 knockout cell or PXB cell. Further, we show that AGAP2 had high gap activity toward ARF1. ARF1, that is a key molecule for retrograde trafficking in Golgi apparatus. We find that DOC11 exhibit GEF activity toward ARF1, and co-IP analysis show that ARF1 co-precipitate with DOC11 and AGAP2. So these results indicate that DOC11, AGAP2, ARF1 form complex that facilitate retrograde trafficking of HBV. So bereferidine A, BFA, the inhibitor of ARF1, potentially block retrograde trafficking of HBV from early endosome to trans-Golgi network. In fact, CCC DNA level was significantly inhibited by the BFA treatment. So thus, the DOC11 and AGAP2 regulate ARF1 activity to promote HBV retrograde trafficking. So finally, we evaluated the potential utility of DOC11 as a target of HBV in vivo situation. PXB mice was infected with HBV for 8 weeks, and then lipid nanoparticle LNP embedded SI.11 was injected through tail vein every week, total 4 times. And after 28 days, serum and liver HBV was evaluated. So DOC11 was efficiently reduced by the LNP SI.11, and serum HBV DNA and HBs antigen and HB antigen was significantly suppressed, as shown in the slide. So in the liver tissues, the expression of HBs antigen and HB core antigen was reduced by the suppression of DOC11. And more strikingly, the CCC DNA in the liver was substantially replaced as observed in southern protein by suppressing DOC11. So these results indicate that suppression of DOC11 could be a good candidate strategy for achieving a functional cure of HBV infection. So in conclusion, there is a non-canonical retrograde trafficking of HBV from early endosome to trans-Golgi network, endoplasmic reticulum, and to the nucleus that potentially avoid lysosome degradation of HBV and result in more CCC DNA formation. DOC11 and its partner AGAP2 regulate ARF1 activity to facilitate the HBV retrograde trafficking. SI-DOC11 significantly reduce the CCC DNA in HBV-infected PXB mice. Therefore, DOC11 could be a druggable target for elimination of HBV infection in the liver of chronic hepatitis B. Here is a collaborator. Thank you for your attention.
Thank you very much, Professor Honda. Okay, we have some questions from the audience.
G'day.
Yes, please go ahead.
Hi. Great talk. I have a question about whether you know whether the genes that you're knocking down or altering have any effect on cell mitosis, because we know that mitosis of infected cells results in 2 uninfected daughter cells.
So far in using a cell line, so SI-DOC11 does not affect the cell division so far. Yeah.
Thanks.
Any other comment or questions? Okay, please. Please be using microphones.
Nice work. I have a quick question. Just your slides show that if the DOC11 will help eliminate the cccDNA, and if the cccDNA eliminate, where does the S antigen come from?
Oh, I mean that if we can eliminate the cccDNA completely, maybe the infectious replicating HBV could be eliminated. However, S antigen could be integrated into the host genome, right? So maybe S antigen somehow continue to be positive. Is that okay?
Okay, so if they have you detach the E antigen at the same time, if I think if the E antigen is negative, all the S antigen will come from the integrated DNA. So have you detached the E antigen when the cDNA is eliminated from the
that would be
cells?
a good question. But so far, we actually we in vivo in PXB mouse, so it's not a chronic infection model. It's a kind of the what to say 8-week infection. So maybe viral genome will not be integrated, I guess. So yeah, I think your question is very important, but so far we could not know that actually.
Okay, got it. Thank you.
Any other questions or comments?
Dr. Honda, very excellent presentation. Thank you. At first, I'd like to ask you about the clinical setting. It is reported that DOC11 deficiency or mutation of DOC11 cause anemia or immunological dysfunction. What could you tell us the points to be considered when it compounds will be introduced to a clinical setting?
Yeah. So so far, when we started this study, and there are no such report. But last year or this year, early this year, actually, DOC11 have some functional role in the immune cell. So far, our strategy is that we would like to suppress DOC11 only in the liver. So when we utilize lipid nanoparticle, most of the lipid nanoparticle are accumulated in the liver. Also, we are now developing a hepatocyte-specific peptide against the DOC11. So maybe hepatocyte-specific therapy target might be useful. Is that okay?
And one more question. It is reported that turnover of CCC DNA is shorter than previously supposed. It's several weeks to several months for now. And it's because the immature nuclear capsid recycled in the cytoplasm and translocate to the nucleus. In this situation, this kind of nuclear capsid, it's a possibility to use this alternative retrograde trafficking system, or it's used by a canonical pathway.
Yeah, that would be a very good question. So we used HepAD38 cell. It is stably actually integrated in the HBV genome under the CMV promoter, regulated by the TET system. So using the HepAD38, it also do not have the NTCP receptor. So SI DOC11 using a stable knockdown of the SI DOC11 using a lentiviral system. So actually after 20 days or 30 days, CCC DNA is completely gone. So I think this might be a SI DOC11 would interfere the recycling of the HBV. That's our guess. So as you said about that half-life time of the CCC DNA, yeah, actually using a cell line system, it allowed maybe 2 or 1 month, I think, but it's still long, very long. But it's not consist forever. But it would be degraded, I guess. Is that okay?
Thank you.
Any other comments or questions? No? Thank you very much, Professor Honda. Nice work.
Okay, thank you very much.
Now we move to the last speaker of this session, entitled the analysis of HBs antigen and cccDNA reduction by nucleotide analog and pegylated interference to combination therapy by Professor Tsuge from Hiroshima University. Professor Tsuge.
Good afternoon, everyone. I'm Masataka Tsuge from Hiroshima University. First of all, I appreciate Professor Shina for having given me a great opportunity to talk about my research. So today I'd like to present a talk with a title, Analysis of HBs antigen and CCC DNA reduction by nucleotide analog and pegylated interferon combination therapy. I have nothing to declare for this study. This is the current Japanese guideline for the treatment of chronic hepatitis B. This guideline indicated two goals of antiviral therapy. Although most of chronic hepatitis B patients can achieve the short-term goal by nucleotide analog therapy, but it is difficult to achieve the long-term goal of HBs antigen loss if nucleotide analog therapy was continued for several years. When we analyze the cumulative HBs antigen loss rate in chronic hepatitis B patient with nucleotide analog therapy, only 2.2% of patients achieve HBs antigen loss during nucleotide analog therapy. On the other hand, several reports indicated greater HBs antigen loss or reduction rate by interferon therapy. This is a famous report that indicated additional antiviral effect of interferon add-on therapy in chronic hepatitis B patient with nucleotide analog therapy. As shown in this graph, HBs antigen loss rate with 48 weeks of tenofovir plus PEG interferon combination therapy was significantly higher than with the other therapies. So this might indicate that the combination therapy can induce HBs antigen reduction more effectively. I'd like to consider the advantage of nucleotide analog and interferon combination therapy compared with interferon monotherapy. We prepared human hepatocyte chimeric mice with and without HBV infection and treated the mice with interferon. After interferon treatment, we compared gene expression profiles in the human hepatocyte between HBV infected and non-infected mouse livers. Downregulation of interferon responsiveness by HBV infection was observed, and it's the most downregulated canonical pathway was interferon signaling. So how does HBV inhibit intracellular interferon responsiveness? To understand the impact of HBV infection on intracellular gene expression, we performed next generation sequencing using HBV infected and non-infected chimeric mice livers. Gene expression analysis revealed that IL-8 was one of the genes most upregulated by HBV infection. To verify IL-8 upregulation by HBV infection, IL-8 expression was quantified in hepatoma cell line and in human hepatocytes. As shown in the left graph, IL-8 expression in HBV producing cell was significantly higher than in control hepatitis cells, and IL-8 expression was significantly reduced by entecavir therapy. A similar alteration of IL-8 expression was also observed in human hepatocyte chimeric mice liver. We performed further analysis and found that IL-8 transcriptional activation occurred after HBV infection via ER stress caused by the accumulation of HBs antigen in ER. So how does IRH upregulation affect in interferon responsiveness? We generated IRH producing cells by stable transfection of IRH expression plasmid and analyzed interferon responsiveness. Although intracellular interferon stimulated gene, ISGS, were induced by interferon treatment, the induction rate of ISGS in IRH producing cells were suppressed compared with original cells. In fact, immunocytochemistry showed that MXA expression was obviously suppressed in IRH expression producing cells compared with non-producing cells. Thus, we consider that increase in IRH production by HBV infection could suppress intracellular interferon responsiveness, resulting in induction of resistance of interferon therapy. So what shall we do to recover interferon responsiveness clinically? As suppression of HBV replication might lead to reduce intracellular IRH production by the previous experiments, we analyzed serum IRH level in 85 chronic hepatitis B patients with entecavir treatment. Individual serum IRH level varied before entecavir treatment, but the level of IRH gradually decreased, and after more than 1 year of entecavir treatment, the levels were significantly lower at the baseline. Therefore, I hypothesize that chronic hepatitis B patient undergoing long-term nucleotide analog therapy might have improved interferon responsiveness. Although there are two protocols of interferon therapy, add-on therapy and sequential therapy for chronic hepatitis B patients, but it has not been clarified which combination therapy is more effective in achieve HBs antigen and CCC DNA reduction. So we conducted a clinical study and compared antiviral effect among these two interferon treatments. 21 Japanese chronic hepatitis B patients were enrolled, and 11 patients were enrolled add-on therapy, and the other 10 patients were enrolled sequential therapy. Here is a baseline characteristics of enrolled patients. There are no significant difference between two groups. So during PEG interferon therapy, the change in HBs antigen level for each patient is shown in this graph. HBs antigen level were reduced by a median 0.5 log during PEG interferon therapy, and more than 1 log reduction could be observed in 8 of 21 patients. 6 of the 8 patients were treated with sequential therapy, and the remaining 2 were treated with add-on therapy. So it was considered that sequential therapy might be more effective for the reduction of HBs antigen level during PEG interferon therapy than add-on therapy. Does the HBs antigen reduction continue after PEG interferon therapy? This graph shows the trend of HBs antigen level each patient. The alteration of HBs antigen level 1 year after PEG interferon therapy was 0.03 log in median, and the alteration of level was different among individuals. The patient with sequential therapy, HBs antigen loss was achieved 1 of 10 patients, and continuous HBs antigen loss reduction after PEG interferon therapy could be observed in 4 patients. On the contrary, in all patients with add-on therapy, we could not observe HBs antigen reduction after the end of PEG interferon therapy. When we analyze factors associated with HBs antigen reduction by univariate analysis, a lower HBs antigen and level at the start of PEG interferon therapy and the regimen of interference therapy was significantly associated with HBs antigen reduction after the end of PEG interferon therapy. To find the other factors associated with HBs antigen reduction by PEG interferon therapy, we analyzed the alteration of laboratory data in each patient. Then we found cases in which ALT elevation occurred during HBs antigen, during PEG interferon therapy, even though HBV DNA level were maintained low level. We consider that this kind of ALT elevation could be associated with antiviral effect and analyze the association between ALT elevation and HBs antigen reduction. As shown in the left graph, HBs antigen reduction level from end of interferon therapy to 1 year after interferon therapy and maximum ALT level during PEG interferon therapy were significantly correlated. And HBs antigen level in patients whose ALT was elevated to more than 50 during PEG interferon therapy were significantly more reduced than those without ALT elevation. In addition, when we measured serum IL-8 level before and at the end of interferon therapy, we found that cases whose serum ALT level at the end of interferon therapy were lower, significantly associated with greater HBs antigen reduction after 1 year from the end of interferon therapy. On the other hand, when we analyze factors associated with HBs antigen reduction at 5 years after PEG interferon therapy, lower platelet count and a higher ALT level at the start of PEG interferon therapy was significantly associated with HBs antigen reduction. However, no association was found between HBs antigen reduction and interferon therapy. The cumulative rate of HBs antigen loss in cases that received sequential therapy was marginally higher than those with add-on therapy. Can the combination therapy reduce intracellular intrahepatic CCC DNA? Since we could not obtain liver tissues before and after PEG interferon therapy from enrolled patients, we analyzed intrahepatic CCC DNA level using chimeric mouse livers. we generated HBV-infected humanized mice and treated them with peg interferon alone or with nucleotide analog plus peg interferon. After interferon treatment, we compared serum HBs antigen and intrahepatic CCC DNA level. While serum HBs antigen were reduced by about 0.5 log following interferon monotherapy, HBs antigen level was much more reduced by combination therapy. Furthermore, intrahepatic CCC DNA level was also more reduced by combination therapy compared with interferon monotherapy. Therefore, we consider that nucleotide analog plus peg interferon combination therapy could be expected to reduce not only serum HBs antigen and also intrahepatic CCC DNA level. This is a summary of my talk. Sequential therapy might be superior to add-on therapy in reducing HBs antigen level in chronic hepatitis B patient undergoing long-term nucleotide analog therapy. AT elevation during peg interferon therapy and lower islet level at the end of peg interferon therapy might be useful marker for predicting HBs antigen reduction after peg interferon therapy. Although it is hard to achieve CCC DNA loss under current antiviral therapy, the incidence of HBs antigen loss and CCC DNA reduction rate might be increased by revising the usage of current therapies. This is our collaborator. Thank you for your attention.
Thank you very much for excellent talk. Is there any questions from audience or internet? I have one question. You find some difference between entecavir and TDF? What is the rationale for finding some
when we
difference?
analyze the clinical trial, we don't have enough patient by entecavir tenofovir therapy, so we could not compare the effect between entecavir and tenofovir.
But your experimental results showed
yes.
There was a difference.
When we analyze chimeric mice, tenofovir is better than entecavir, but we don't know that that effect can be affect the clinical trial.
Okay, thank you. Any questions, comments?
Please, if upregulation of IL-8 suppress interferon responsiveness, at that time, serum IL-8 levels at the beginning of peg interferon therapy is supposed to be a better marker than at the IL-8 level at the end. But your study shows the significance is latter.
When we measured the IL-8 level, serum IL-8 level before peg interferon treatment, we could not observe the difference of HBs reduction. But at the end of interferon therapy, we can observe the difference of HBs antigen reduction. So if we can predict before interferon therapy, but from my research, we cannot indicate before interferon treatment using serum IL-8 level.
Thank you.
I have other questions about IL-8. And is there any genetic host immunology backgrounds? Is host genetic backgrounds like SNPs of IL-8?
I have never analyzed that SNP.
It could be possible because you can find some
Yeah.
as for interval responsiveness.
Yeah,
out of
we
genetic-
have many genetic data, but we have never analyzed that point.
Okay, thank you. That's future questions. Is there any comment? No? Okay, Professor Sugi, thank you very much for excellent
Thank
talk.
you.
I think, is there any question or comments from the audience about this regarding past 3 presentations? Or maybe, do you have any comments or something? No, no further? Okay. And then I think the topic is very heterogeneous among the speakers. So I don't think the panel discussion is very useful. So if there are no comments or no other suggestions, I'd like to close these sessions. Okay. Thank you very much for the presentation.
Thank you very much, doctors. We'll now have a coffee break. We will start the next session at 3:30 p.m. We'll start the next session at 3:30 p.m.